Here's a representation of a protein in mole view. You can do this yourself because remember, mole view is free. The website is moleview.org. And this particular protein that we're looking at is amylase. So over here in this bar, you can just type in, search the name of any protein. And if it has it in the database, it'll come up and you can look at it. I picked the amylase protein because it has a combination of alpha helixes and beta pleated sheets, so you can look at how it's represented. Now, what you're looking at here is a really simplified representation of the structure of this protein. But in this simplified representation, the alpha helixes are being shown in purple. So you can see there that purple, magenta, twisted shape. And this protein, amylase, has got quite a few alpha helix portions in it. So if I rotate it around, you can see there are multiple parts where it takes the form of an alpha helix. And the beta pleated sheets are the portions that are shown in blue, and there's quite a few of those as well. And you can see that they're kind of side by side with each other. It's not really showing the kinked, folded, pleated part of it, but it is showing the flatness of the beta pleated sheet. And so I feel like this is a great one to look at because it gives us a chance to really see that most proteins have both alpha helixes and beta pleated sheets, and they're kind of spread out within the protein. And then you've probably noticed that there's a lot of kind of like grayish white ribbon looking segments, the skinny lines that don't have anything at all. So those are the portions of amino acids that don't have secondary structure. They're just kind of a random collection of amino acids. And you might also be noticing in this guy, I don't know if I can really zoom in on it, there's like a couple random molecules in there too, just hanging out, random atoms hanging out. So as you can see from amylase, from this particular protein, that as it twists itself into alpha helixes and folds itself into beta pleated sheets, it also kind of scrunches itself up into a wad, or, it's not really a sphere, but it just kind of munches itself up into this shape. And this mushed up, crunched up shape is the next level of description of a protein. So number 3, the next way that we would describe the shape of a protein. And this is called the tertiary structure. The tertiary structure is what just, kind of squishes the whole thing together. It's also a 3-dimensional shape. So it's a 3-dimensional shape, just like the alpha helix and the beta pleated sheet. It is compact, and it is complex, and it is stable in terms of energy. So when the protein squishes and compacts itself into the tertiary shape, it helps to stabilize it. And it's more compact and more complex and more stable than the secondary structure. So more than our secondary structure. The cause between the tertiary structure, so if we go back up and kind of remind ourselves, for the secondary structure was caused by hydrogen bonding between amino acids, The tertiary structure is caused by interactions, different types of interactions, caused by interactions between the R groups of our amino acids. And because there are all different kinds of R groups, we have the acidic, we have the basic, we have the polar, we have the non-polar, caused by interaction between the R groups of amino acids. There's a whole bunch of different types of interactions that could arise. And we're going to lump those types of interactions into three different types. So one type is the hydrophobic type, which is sometimes called the London or the dispersion, or the London dispersion, not dispense, dispersion. So hydrophobic type, London dispersion London dispersion force, anything along those lines. This is what this is referring to. This is an interaction that takes place between the R groups of nonpolar amino acids. And they don't really do anything in particular other than they just sort of clump themselves together. So it's between the nonpolar R groups of amino acids. It is caused by the R groups clumping together or clustering together. So they're not hydrogen bonding with each other. They're not really doing anything other than just sort of getting together and hanging out together. And they're driven to do this to get away from the external environment of the protein, which is usually water-based or aqueous. So these nonpolar groups are repelled by the aqueous external environment of the protein. So it's caused also by the R groups moving away or migrating away from the aqueous, water-based, external environment of the protein. And if this sounds vaguely familiar to you, this is really similar to the concept of the way soap molecules work, or the way the cell membranes are put together when we have these lipids that have these hydrophobic, was the word we used, hydrophobic tails that are trying to get away from the water, and they just cluster themselves on the inside to try to protect themselves from coming in contact with the aqueous environment. Second type of interaction between the R group is called hydrophilic. This is also sometimes called electrostatic. And this is basically the opposite of the hydrophobic interaction. So this is going to be between the R groups of polar amino acids. And remember that includes, polar amino acids includes acidic and basic amino acids as well. So it's between the R groups of polar amino acids, and it's happening for the opposite reason. So it is caused by these polar R groups clustering together and they're going to cluster together on the surface. They're going to create the surface, the exterior of the protein, in contact with the aqueous external environment. And then the third type, the last type, is very specific, and this is called a disulfide bond. And it also is called a disulfide bridge. And this is just the hydrophobic and hydrophilic are pretty, generic. Any polar or any non-polar amino acids clumping together. The disulfide bond is very specific. It is between two cysteine amino acids. And that's it. And as a reminder, cysteine looks like this. CH2SH. And so if we have, you know, a trail, chain, whatever, of amino acids, and then down here we have another amino acid, which I'm going to draw upside down, NH3 plus, COO minus, CH2, SH. And if these two cysteines end up coming near each other, sort of in contact with each other, they can interact in this way and form a connection between the two of them, which we call a disulfide bond or a disulfide bridge, because it's this connection between two sulfur atoms. So that's a very specific, particular connection. So this third type, the tertiary structure, the squishing together of the amino acid, is what we see here with the amylase. Now, we don't actually see a representation of how these R groups are interacting with each other, but if we play around with this structure, if we play around with the way that we're looking at it, so let's say that we want to see, we want to see all the bonds in this guy. Whoa, there they are. And so if we wanted to zoom in, this is really overwhelming, right? There's a lot of atoms in this protein. If we wanted to spend some time trying to analyze this, looking at the R groups that are now visible to us, so we are able to see them, we would be able to get a picture of some of these tertiary interactions. You know what I'm focusing on right here, right now? So the standard color of sulfur is yellow. And do you see those yellow lines that are kind of coming into view? I don't think there's a way that I can like click on them and highlight them, but now they're up here at the top. I'm going to try to touch them. I can't really touch them. So there's this yellow thing right here that is up at the top that's moving around. That is a disulfide bridge. Those are two sulfurs that have connected together and formed that tertiary interaction. There's quite a few of them in this amylase molecule, which is pretty awesome. So yeah, that's really overwhelming to see it like that. There's also a fourth shape for our proteins that not all of them have. And the fourth shape is called a quaternary structure. And the quaternary structure exists only when we are dealing with a protein that is made of multiple proteins. So this would be for two or more, we call these protein subunits. So we get two or more individual proteins that come together, and those subunits are held together by the same type of interactions as the tertiary structure. So they're held together by the same interactions as a tertiary structure. Hydrophobic, hydrophilic, and disulfide. So what exactly does this, I'm so excited I can't even spell right. What exactly does this work or look like? So this would be like we have one protein, here it is, that's my best way to draw a protein. And then we have a second protein, here it is, blah. And then those two, and these are individual proteins, they have their own sequence of amino acids. There's no connection between them in terms of connecting them through a primary structure. Their amino acids are not peptide bonded to each other in any way. So then these two individual proteins just sort of come close together, and they just cluster together. They don't actually bond, except for maybe through a disulfide bridge here and there. They're not actually bonding together. And the classic example of the quaternary structure is hemoglobin. So I'm going to see if I can quickly change this to hemoglobin. Hemoglobin contains 4 individual proteins that are clustered together. And it's just, it's just really hard to see this in this sort of notation because there's so much going on here. Proteins are so big and so complicated. So I don't know if I can get this set up in a way where you can see the individual structures. Like right now where I'm looking at it down on the bottom of the screen, you can see the end. In white, you can see the end of one of the amino acid chains. And then I'm going to rotate. So over to the left, if you look over to the left, there's a yellow ball. There's a yellow ball up above, and there's also a yellow ball over to the left. Over to the left, there's another end of an amino acid sequence, which I'm trying to rotate it so that it's kind of comes into the middle. So you can if you twist this around a lot, you can find the individual subunits of this protein. The easiest way to see the subunits of these proteins are with the heme molecule that's in the center of each hemoglobin. So every individual hemoglobin has a heme. The heme is a molecule, which I'm going to try to get into a good view and then zoom way in on it. So the heme molecule is going to be represented by ball and stick. And here's one right here. It's come into view. It's got a white atom in the center. It's got 4 blue atoms attached to it. Then it's got a lot of gray stuff around it. That is an individual heme. So if we zoom out, you can see that this protein has got a total of 4 hemes. Each heme is in the center of its own protein. So from that, we can imply that there are 4 individual proteins here, and then they're just being sort of clustered together to make this overall massive molecule. And if we want it to get real crazy, we're going to look at the bonds. So this is depending on how big of a nerd you are, maybe play around with this. Look at different proteins. Up here you can search different proteins and see what they look like.